Cells moving, meeting, and moving away again. Movement achieved by the ruffling of the cell membrane. This shows the membrane as a mobile and very dynamic structure, but a freeze-fracture image of the cell reveals a structured and rather static image of the membrane surface. And a conventional electron micrograph, here two membranes closely opposed, shows each to have a tram-like appearance, a trilaminar structure. Now, biochemistry tells us that the membrane is composed of proteins and lipids. Can we construct lipids and proteins into an arrangement that will fit with these images? Whatever arrangement we construct must take into account the functions of membranes. Membranes have to be permeable to allow nutrients to enter the cell, and yet be impermeable to maintain the integrity of the cell. To solve this paradox, let's look at the arrangement of the principal macromolecules. Dr. Dennis Hayden, University of Cambridge.
Hydrocarbons don't dissolve in water. The water's covered for the purposes of television. Now, phospholipids, for this reason, don't dissolve in water either. They form aggregates. In a biological membrane, these aggregates take the form of a bimolecular leaflet. That is, a leaflet just 2 molecules thick. Just 2 molecules. Now, why 2 molecules? The answer is to be found in the structure of the phospholipid molecule. This molecule has a polar group and 2 non-polar chains. The polar group tends to position itself in the aqueous phase, and the non-polar chains tend to associate together so as to minimize their contacts with water. Now, if we look at the dimensions of the molecule, we find that the cross-sectional area of this polar group is very similar to the cross-sectional area of those 2 chains. This makes the molecule roughly cylindrical in shape, rather like this beer can. And it's because of this cylindrical shape that the molecules compact very easily side by side. The beer cans illustrate the point very well. Here we have a monolayer of phospholipids, the polar groups and the chains. Now, as you see, the ends of the chains are still exposed to water, and so this structure is not ideal. However, if we put another monolayer back to back to the first one, so that the chains are in contact with each other, we can completely exclude contacts of the chains with water, and we have pretty well the ideal structure. In this structure, the chains fill all the available space, and in biological membranes at ambient temperatures, the chains are in a liquid state.
Now, just such an arrangement is likely to produce a significant barrier to the movement of small, useful substances, such as ions, amino acids, and sugars. The proteins that are concerned with permeability, we need to look at their arrangement in the membrane. The original idea of Dowson and Danieli had the lipid bilayer sandwiched between continuous smears of protein, but Singer and Nicholson had other ideas. Professor John Singer.
Now, what we've learned about proteins has come largely from studies of X-ray crystallography of water-soluble proteins in a crystalline form. And there it's become clear that the molecules of proteins are globular, compact structures, very complex conformation, and not single alpha helices at all, but convoluted into some shape in which the hydrophobic amino acids are intercalated largely into the membrane interior, and the hydrophilic amino acids are largely on the membrane exterior. This maximizes simultaneously both the hydrophobic and the hydrophilic interactions. And the question was how to relate this kind of structural information to the problem of membrane proteins. Now, I entered this field about 1960. My background is a protein chemist. And the question was how to relate these problems to the problem of membrane protein structure. The solution was obvious to most protein chemists if they had addressed themselves to this problem, and that was to have a protein molecule organized as an amphipathic structure in which the hydrophilic amino acids would be largely localized to the exterior surfaces of the molecule where the protein would come into contact with the water, and have the hydrophobic amino acid residues localized to the interior part of the molecule where it would come into contact with the hydrophobic fatty acyl chains of the phospholipids. Now, what happens if we try to place a molecule of that structure into the lipid bilayer of the membrane? That can be visualized in this model. What you see resulting is that the hydrophobic surface of this amphipathic protein is in the interior region of the membrane where the hydrophobic fatty acids are located, whereas the hydrophilic exterior of the protein is placed out into the aqueous environment. And if this protein is one which spans the thickness of the membrane, then there are these two hydrophilic surfaces exposed at either side. Now, if we look at the top surface of this model, what is seen is that this represents a kind of mosaic structure in which the individual proteins are randomly disposed in the lipid bilayer. In particular, if the lipid itself is in a fluid condition, then there are no restrictions holding these proteins in any particular place in this plane, but rather they should be freely mobile in the plane. Now, this picture of membrane was quite unexpected. Most people who thought about membranes at the time thought of them as rigid structures in which the proteins formed some kind of a lattice which was quite immobile. But around this time, Fry and Edidin performed a very remarkable experiment. It is possible to attach fluorescent labels to the surface proteins of living cells. A label of one color was attached to human cells and of another color to mouse cells. The cells were then made to fuse together. Initially, the labels were separate, but within a half hour, they had completely intermixed. Another problem that this model addresses is the problem of permeability and transport. By a minor variation of the protein structure that we've been talking about, we can, for example, envision that a protein exists in the membrane as a kind of subunit aggregate with a water-filled channel running down the interior portion of the membrane through the membrane to the other side. The point here would be that the channel would be lined by hydrophilic residues of the protein molecule so as to satisfy the thermodynamic interactions involved, and the small molecule ion or other hydrophilic ligand could then traverse the membrane from one side to the other. One of the main reasons to have a protein being transmembrane, I think, is to be involved in these transport processes. The existence of proteins that span the entire thickness of the membrane is most aptly demonstrated by freeze-fracture experiments which have been pioneered by Daniel Branton of Harvard University.
Very often we can actually see transmembrane proteins by freeze fracturing. For example, in the case of the red blood cell membrane, which we know is very active in anion transport, one can see a large number of bumps on the surface of the membrane, and in fact it's clear that these bumps do represent the transmembrane proteins that serve as anion channels. In contrast, on this photograph, for example, of myelin membranes, we see a relatively smooth surface. These faces are representative of membranes which are inactive in terms of transport. In fact, they serve primarily as insulators around myelin. But the differences aren't all that clear-cut. In many membranes, a protein traverses through the lipid bilayer as a alpha helix only once. And then it's too small to be visualized by freeze fracturing, so we see nothing. We don't see any bumps, even though there is a transmembrane proteins. Nonetheless, because transmembrane proteins frequently go through the membrane more than once, freeze fracturing serves as a very useful way of looking at these transmembrane proteins.
So far then, our model looks like this. Proteins lying in the lipid bilayer, floating freely in a lipid sea. But what's the importance of this fluidity? One of the key functions of membranes is to communicate events happening outside the cell through to the cell interior. And many of the proteins in the membrane act as receptors for signal molecules like hormones. And many hormones, in fact, exert their cellular effects by activating an enzyme, adenylate cyclase, which is in fact well buried into the membrane on the internal face. Activation of adenylate cyclase produces cyclic AMP from ATP, and it's cyclic AMP, this second messenger, which initiates the cellular response. Both proteins, receptor and adenylate cyclase, appear to float freely in opposite sides of the membrane. Random meeting elicits no response, and they drift apart. But when the hormone is present, and this binds to the receptor, it activates the receptor such that a subsequent meeting of the hormone receptor complex with adenylate cyclase now results in the production of cyclic AMP. But in the same way that fluidity permits the coming together of proteins in the membrane, it can also take them away from one another, and this would not be very satisfactory in terms of function. It seems, in fact, that not all parts of the membrane of a cell are equally fluid. There are areas in which fluidity is controlled, such as the junctions between cells, the so-called gap junctions, and the points of interaction between the cells of the nervous system, the synaptic junctions.
That's a bivalent chromosome being stretched by means. What you can see in this film is a bivalent, that's a bivalent chromosome, being stretched by means of a very fine microneedle. When the microneedle is removed, the bivalent springs back to its original shape and position. That film was shot by Dr. Bruce Nicholas. Now, the thing that occurred to me when I first saw that film was, how do you do that experiment? After all, a chromosome is only about 10 microns long, which means that the length is about 1/10 of the thickness of a human hair. So how do you get a microneedle and manipulate chromosomes like that? Well, another thing, and this is really what the program's about, is the very fact that you can stretch that bivalent with a needle, and when you remove the needle, the bivalent springs back again, indicates that the bivalent must be attached to some structure in the cell. Now, in that film, you can't actually see the structure, but for evidence of that sort, and as you'll see from other evidence, you can postulate that during mitosis and meiosis, chromosomes are attached to some physical structure in the cell, a structure generally known as the spindle. And that's essentially what the program's about. The movement of chromosomes, in particular, during mitosis. But before we get involved in the detailed mechanics of how mitosis operates, let's actually just broadly recap the process again. And to do this, we'll use a film made by Dr. Andrew Beyer of mitosis in the African blood lily. Right, here's the resting or interphase nucleus, where you can't actually see any distinct chromosomes. Now, as we come into prophase, you can just begin to see chromosomes appearing as condensed threads. And remember that each of these threads, in fact, consists of 2 chromatids. Now, in prometaphase, they organize and they move up towards the central equator or metaphase plate. Now, this movement of the chromosomes during prometaphase towards the metaphase plate is one of the two stages of mitosis that we're going to consider in detail in this program. The other stage is where the daughter chromatids are distributed to the opposite ends or poles of the cell, anaphase, which happens next, or almost next, because first we're going to pick up the process more or less where we left it, where the double chromosomes are lining up on the metaphase plate. Now you can see them more or less lined up on the metaphase plate, and they'll just begin to split into daughter chromatids now. Right, here they go. They're in anaphase. They move off towards the opposite poles of the cell. And in late anaphase, you'll begin to see, there it is, the new cell wall material forming in the middle, and the chromosomes will become less distinct as each of the two new nuclei go into interphase. Of course, that mitosis takes several hours, but we're using time-lapse photography. In fact, chromosomes move at just a micron or two a minute. That's much slower than the movement of an hour hand on an ordinary watch. Well, let's watch the whole process again and look for, in particular, the two stages we're interested in. Right, prometaphase, one of the stages we're interested in. And that's the other, anaphase. Now, when you see a piece of film like that, the thing that strikes one is the accuracy, the fidelity of the process. And of course, this is the way it has to be, because the whole purpose, as it were, of the process is to distribute equally the sets of chromosomes to the two new daughter cells. So if you're thinking in terms of mechanism, I don't think it's much use imagining that the chromosomes are sort of like sausages swimming in a sea of porridge, and the daughter chromatids are distributed by random movement into two new daughter cells. I think the thing must be much more structured than that. And because of this, from very early days of studying mitosis, way back in the 1870s, people started to think in terms of physical structures in the cell which helped organize this distribution of the chromosomes to the two new daughter cells. In fact, if we look at these drawings produced by the early microscopist Theodore Boveri in 1887, we can see he's drawn these physical structures, which he termed spindles, which he considered as being involved in mitosis. Now, in this first sequence of pictures, where we deal with the movement of the chromosomes towards the metaphase plate, you can see how he's indicated that the chromosomes are attached to the spindle rays or fibers, suggesting in some way that they organize the movement of the chromosomes towards that metaphase plate. Now, in the second sequence, where we're looking at the distribution of the chromosomes, again it seems as if the chromosomes in some way move up the rays of the spindle, in this case, towards opposite poles of the cell. Okay, so from the work of Boveri and others, we can accept that spindles exist. But if we're to understand how they operate during mitosis and meiosis, we need to know more about them. First, it would be useful to know something about the chemical composition of spindles. Then we really need to establish that chromosomes are attached to them, and if so, how? Finally, are spindles really involved in chromosome movement, and again, how? So what we're going to do in the remainder of the program is take a look at some of the techniques that have been applied to these questions, and briefly, what answers we now have. Right, let's look at how one sets about investigating the composition of spindles. First of all, you need to be able to isolate spindles. And this was done in the 1950s by Mazier and Dan. To do this, they obviously had to choose a material that one would suppose had a large amount of spindle. And the material they chose was recently fertilized sea urchin eggs, where there's a great deal of cell division and so a great deal of mitosis. By treating these eggs with a mixture of hydrogen peroxide and a detergent, they were able to dissolve away the basic egg material, leaving the complete spindle structure with chromosomes intact. Well, I think you can see what they isolated looks rather like the things that Boveri drew, where you've got the chromosomes attached to this spindle. Techniques like this have established that spindle is in fact composed of protein subunits, which we'll consider a bit later. Now, no, interesting, this still doesn't prove that these spindles are really involved in the mitotic or meiotic mechanisms as such, nor does it help us establish how they might be involved. This is where I'd like to return to the film we opened the program with, the very beautiful film made by Dr. Bruce Nicholas, where, as you saw, bivalent chromosomes can be stretched or manipulated with a microneedle. But I'd just like for a second or two to consider how, in fact, you do this experiment. First of all, you need a very fine microneedle. And in fact, the microneedle that Nicholas uses is only 0.1 microns in diameter. That's something like 1/1000th of the thickness of a human hair. The other thing you clearly need is some very accurate, sensitive form of micromanipulator, whereby you can make rapid, small movements of the needle so as to pinpoint exactly what you want to move without damaging the cell. Now, most conventional micromanipulators are just not good enough. They're not sensitive or accurate enough. Almost all have some degree of whip or backlash. In other words, there's a delay between operating the control and movement in the needle. Slack in the system, if you like. And in fact, the micromanipulator that Nicholas used is one based on a design by Gordon Ellis, which depends for the deflections on the piezoelectric effect. Now, the piezoelectric effect is a means whereby applying measured electric currents to certain crystalline materials, you can produce accurate small physical distortions of those materials. The device is used in conjunction with a microscope, and the control of the movement of the microneedle is by means of a small joystick, which you can see there. Well, I don't want to go further into this particular technique. In fact, in the last few seconds, I've told you virtually all I know about it. But needless to say, here again, there's one example where the sort of experiments one wants to do depend very much on the development of a particular technology. In this case, the technology of accurate micromanipulators. Well, okay, we'll have a look at the Nicholas film again. Now, this is the sort of field you'll be able to see. And look closely at this sort of ghostly streak here. That, in fact, is the outlines of the glass microneedle. And when you see the film, you might just be able to make out something like that, occasionally darting in and out as the chromosome stretched. Now, the chromosome we're interested in is that one in the bottom right-hand corner of the cell in the center of the field. Now, wait and watch for the needle. There's the needle going in. And you can see the chromosome being stretched out to many times its normal length, like a pair of braces being pulled. And when the needle's removed, you'll see it spring back to its original shape and position. Now, the needle's out now. There it goes. Now, the needle hasn't damaged the cell because the cell goes into anaphase. So chromosomes are attached to the spindle during meiosis and hence mitosis. Of course, it's all very well establishing that during mitosis there is a structure called the spindle to which the chromosomes are attached. But that in itself tells us virtually nothing about how the chromosomes move towards the metaphase plate, and then having split into the two daughter chromatids, how those chromatids move towards opposite poles during the distribution phase. There's lots of questions we like to ask. For example, exactly how are the chromosomes attached to the spindle? How do they move towards the metaphase plate? And then having reached the metaphase plate, why do they stop? And above all, what's the actual mechanical force that moves the chromosomes around the cell? Is it the chromosome itself which provides the motive force? Or is it perhaps movements of the spindle? Does the spindle contract or expand, thereby moving the chromosomes to different parts of the cells? Does the spindle flex? It's all these sorts of questions which we'd like to answer. Now, in fact, virtually none of the answers are definitely known. But what we're going to do in the rest of this program is look at some of the ideas and some of the evidence concerning them. Some of the evidence is gathered from cells in mitosis, some from cells in meiosis. There are, of course, important differences between these processes, as we've indicated in the broadcast notes. But broadly, for our purposes, the role of spindle structures and chromosome movement, it's probably okay to collate evidence from both processes into a sort of general scheme. Now, because very little is actually known about the way in which chromosomes move and how the spindle organizes this, many different types of hypotheses of chromosome movement have been suggested over the years. For example, take a look at this hypothesis suggested by Lilly round about the turn of the century. It's based on the idea that chromosomes move by electrostatic forces, and spindles aren't really involved at all. You see, he's got the cell membrane here, polarized, and the nuclear membrane, supposed to be an interphase nucleus, also polarized. Now, as the cell goes into mitosis and the nuclear membrane breaks down, he suggests you get a redistribution of the sort of electrostatic forces, rather now like iron filings in a magnetic field. And then because of this, you get another redistribution, and the chromosomes, these U-shaped structures here, he suggests are both negatively charged and therefore repel each other towards opposite ends of the cell. Then there's another hypothesis suggested some 30 years later by Schrader. Here spindle is involved, though the idea is that there are convection currents in the spindle which help move the chromosomes around the cell. Well, that's just a couple of hypotheses that have been advanced to explain chromosome movements, and it so happens neither of them is given much credence these days. There are many more in the same sort of category. What we want to do now is look at a little more hard evidence about how chromosomes move. Both the hypotheses we've just seen were really guesswork, in one case based on the existence of spindles, in the other merely based on the places chromosomes could be observed to move during mitosis. First of all, what we'd like to know is exactly how the chromosomes are attached to the spindle, and how any such attachments behave during chromosome movement. What we need is a technique whereby we can observe spindles in living cells during the process of mitoses and meioses. We're not so interested now in looking at the chromosomes during these processes. Those we could see in the films we saw earlier, but we couldn't see spindles in those films. You needed to fix the cells, kill them to see spindles. Now, fortunately, this is no longer true, and it's basically due to the development of a very sensitive polarizing light microscope in the 1950s by Shinya Inoue. You see, fortunately for us, spindles are composed of regularly arranged protein subunits. If you take a structure which is composed of regularly arranged units of this type and put it in a field of polarized light, then it exhibits a phenomenon known as birefringence, which basically means that when the plane of polarized light is one way, the material might be oriented so that it appears dark in the field. Whereas if the material is oriented at right angles to that, it will appear light in the field. You can see the sort of dark appearance of this structure here at one orientation, and again a spindle type thing appearing light at this orientation. So depending on the plane of polarized light, you can observe spindles which either show as light objects or as dark objects in the field during cell division. And using this technique of polarized light microscopy, Shinya Inoue has made a time-lapse film of division in certain grasshopper cells. Okay, you can see several cells during division here, and those sort of Chinese lantern-type structures are the spindles. There's one showing light in the field, and there we've got one showing dark, again, depending on their orientation. Now, most of these at the moment are in metaphase, with the chromosomes along the center. And watch as they go into anaphase there. You can see much of the birefringence moving off towards the poles of the cells. Now, this birefringent material appearing now as sort of parallel streaks is in fact due to mitochondria, which show strong birefringence late in anaphase or early telophase. Now, let's take a closer look at this, again in some cells during division, this time in plant material, again by the same technique of polarized light microscopy. Okay, you're getting to see the whole spindle structure here. Again, the thing is in metaphase. You can just about make out the chromosomes. And look closely as the chromosomes move off towards the poles. See how much of the birefringent material is actually going with them towards the poles of the cell. Now let's have a look at it again. Into anaphase, birefringence. A lot of it is going with the chromosomes towards the poles. So what we can see in those films is that indeed you can see birefringent material which looks like the spindles that Boveri drew. But much of this birefringent material actually travels with the chromosomes during anaphase towards the poles, which suggests that some part of the spindle structure, or at least some birefringent material, moves and is attached to the chromosomes. And evidence of this type has led to the idea that in fact there are at least two basic types of birefringent material during mitosis and meiosis. You've got the continuous spindle rays, which we've drawn on this board here, and these, of course, would appear birefringent. We've got just a few of these continuous spindle rays drawn in. There'll be many more in the cell, but we want to keep it fairly simple. As to these white dots, well, don't worry about those. We'll come back to those in a second or two. So one form of birefringent material you see in the films are these continuous spindle rays. Now, of course, you also saw birefringent material which moved with the chromosomes. And this is believed to be due to so-called chromosomal fibers or chromosome tufts. like these things here. Now here's our chromosome with its 2 chromatids joined by the centromere, and the idea is that these tufts come out either side of the centromere. Well, let's just think about another chromosome, say on the metaphase plate, which is along here, and I'll just build up the tufts a bit. You'll see why in a second or two. Now, one idea is that these tufts are the things that attach the chromosome to the actual spindle rays. So in the Nicholas type experiment, where you pull out this chromosome, or a bivalent chromosome, with a needle, it's these tufts that keep it attached to the spindle rays. Now, in a way, there's done a whole series of experiments which suggest that these tufts are composed of subunits and that the tufts themselves aren't particularly stable. You see, if you treat the cells with a variety of chemicals, you can destroy the birefringence, the tufts vanish. If you then remove the chemicals, within a matter of minutes in some cases, the birefringence, due to the tufts, re-establishes. So in a way suggests that in the cell, there's a pool of subunit material, and that's what these dots represent, and these subunits go to build up the tufts. And so there's an equilibrium between the subunits and the assembled tuft material. Disturb the equilibrium, you either get more subunit and less tuft, or the other way around. Well, so what? Well, in a way, has used this idea, this basic idea of the equilibrium, to suggest a way in which chromosomes might move during meiosis and mitosis. So we've got a chromosome on the metaphase plate, but let's go back a bit and look at one in prometaphase. Now, you probably noticed I've whipped off one of the block of tufts from this end, so they're longer on this side than they are on that. And the reason's fairly obvious from this diagram, because in a way believes there's a greater pull of the subunit material towards the centre of the cell, the metaphase plate region, less out towards the poles. So the tufts pointing in towards the middle will be in this high concentration region, therefore more material will assemble to give tufts. They'll be longer than those out in the low concentration region. Now, because the tufts are longer on this side, you'll get a greater pull, at least that's the idea, and so the whole thing will be pulled down towards the metaphase plate. And as it's moving down towards the metaphase plate, this end will be coming into a higher concentration of subunits. And so gradually, though I'll do it all in one go, the thing will build up and you'll get longer tufts. Now, when the thing, which is a continuous process, gets down to the metaphase plate, the two ends should be about equal length because they've both been pointing out into similar concentrations if this whole thing's symmetrical. When it gets there, since they're equal length, you get equal pull, the thing balances, the chromosome stops on the metaphase plate. Now, the next question is, what happens before anaphase? Well, one thing that must happen is that these two chromatids separate. Presumably the gluing substance here dissolves, two chromatids separate. Now, each of them has got its own set of tufts which point to one or other of the poles of the cell. And so each chromatid moves off towards its pole. Now, what's going to happen to the tufts? Well, as they get nearer the poles, they move into regions of lower subunit concentration. And so the nearer they get to the poles, the shorter and shorter the tufts will get. Well, there's a prediction in the model. If the equilibrium does exist as such, then the nearer the chromatids get to the poles, the shorter their tufts should become. And I think you'll see evidence for this in the next sequence of film shot by Dr. Inouye and Bayer. Okay, there are the chromosomes lined up in metaphase, and see the birefringence. Now watch as they move off in anaphase. There you can see underlying spindle birefringence, and look at the chromosomal tufts. They're getting shorter as the chromosomes move near the poles. They're now very, very short. And here you can see the underlying spindle birefringence still clear. Okay, so I think those films substantiated that the tufts do get shorter as the chromosomes move towards the poles. But what does that actually tell us about the mechanism of chromosome movement? You see, we still really need to know what the interaction is between these tufts and the continuous spindle rays. So let's just for a moment look at some of the suggestions that have been made. Imagine we could have a look at a blow-up of some of these continuous rays. Okay, well here's one way you might sort of visualize the continuous rays. There's 3 rays shown here. And these knob-like things, or cross bridges, are what might exist on each of these rays. Now, if we could also have a look at a chromosome tuft and blow that up, you might see something very similar. Here, for example, will be an individual tuft, again with these knobs or cross bridges. And you might conceive that it sort of interacts with the continuous rays in this way. You've got the tuft on the chromosome with its cross bridges interlacing in some way with the knobs or cross bridges on the continuous rays. Now, because of that interlacing, there might be some mechanism of movement there. We don't know how. I mean, perhaps it's a sort of sliding filament type mechanism, as we discussed in muscle movement, or other people suggest it might be a ratcheting in some way or a zipping up. In fact, there are just about as many hypotheses as there are workers in the field. And really, most of this is very gross speculation. In fact, much of what we say may well be wrong. See, what we're trying to do in the program, basically, is just present some of the directions in which people are thinking. But I suspect before one can really make much more advance in this field, there has to be a further advance in technique.